Hi everyone, welcome to Professor Long's Lectures on Anatomy and Physiology. I'm Professor Bob Long. I'm teaching Human Anatomy and Physiology Part 1 and Part 2. These videos are designed or set up for my students taking my classes. This particular video is set up for my AMP2 class, Human Anatomy and Physiology Part 2. This is the fifth, I believe, video in the sensory physiology series. We're going to be covering the sensations of taste, and we're going to start the eyeball. One of the things I'm learning, this is kind of a crude setup. As you guys know, we're in the coronavirus shutdown or the COVID-19 shutdown. And I traditionally teach face-to-face, but because of our inability to go to campus and meet in large groups, we've moved the classes to online. I'm shooting these with my cell phone, and it's a rather crude setup. I'm not very technologically savvy. So what I've realized is my videos cut off right about 33 minutes, but the phone automatically starts a second video. Why, I don't know, but I'm not losing any content, thank goodness. Nonetheless, I could shoot these with my laptop, but the videos are much darker and grainier. They suffice, and some of the videos may be shot by my laptop. So the ones that are bright and sharp are cell phone, the ones that are dark and grainy are laptop. I'm trying to use the cell phone, but it's much harder and slower to load these into YouTube. I know y'all don't need to know all that, but that's what's going on here. So I'm going to be covering gustation. I'm going to start some of the eye physiology, but we're going to have several lectures over the eyeball because it's a lot of information. If you are in my class, we're going to be on page 10 of the note set. We're going to start gustation, or the sense of taste. And we're somewhere around page 570 in the lecture textbook if you want to follow along in the adopted lecture text. Okay. So gustation is our sense of taste. The receptor cells that detect the gustatory molecules are called gustatory receptors. Okay. Now, the gustatory receptors are located on the little bumps on the surface of your tongue. That stuff, right? So those little bumps are each called a papilla. When you see this term, and you'll see this term a lot, papilla really means nipple, like a little bump sticking out. And so you have papillae on the surface of your tongue. Those papillae have different shapes and different names based on their shapes, and they also function somewhat differently. It is on those little bumps that we have the olfactory receptor, the gustatory receptor organs called taste buds, and within the taste buds are the olfactory receptor cells. They're called taste buds because they look like little buds, like a little rosebud. We're going to go into all the anatomy and detail of that. The sense of gustatory transduction is so close to olfactory transduction, which we just finished, that I'm not going to go into the steps because they're almost identical, or they're pretty close. So, as you know, the gustatory receptor cells are a type of chemoreceptor, just like olfactory receptor cells are, but they do external chemoreception. So the sense of smell and the sense of taste are very closely linked, very anatomically similar, and physiologically similar. Okay. So now, when we look at the surface of the tongue, if we're cruising along the surface of the tongue, there's usually a little fold, and then there's these little structures that stick out. Okay. That fold, by the way, is very often called a taste trench. Okay, like a trench or a ditch in the ground. And the structure sticking up is the papilla. Based upon the shape of the papilla, they get different names. So if the papilla look rather sort of sharp or almost hair-like, the term filly and the word bello and pilli all refer to hair. They're all related words. So we call these filiform papillae. Filiform papillae are hair-like papillae, and they're very narrow and slender, and they're located, a lot of times near the tip of the tongue. Now, one of the things that we know about filiform papillae is they're really not involved in the sense of taste a whole bunch. They do a lot of grip and texture. They can detect textures, they can detect gripping, but they're not really super involved in the sense of taste, and that they don't have a lot of taste buds, if any. There's another one that comes along that looks like a little fold in the tongue, and those are called folate papillae. Folia are folds, so they're little folds on the surface of the tongue. And then we have some that actually stick out like a little mushroom or button, and those are called fungiform papillae. Fungiform in the shape of a fungus. So fungiform papillae look like little mushrooms, or little mushroom buttons sticking off the surface of the tongue. They're nice and smooth and rounded. These actually have quite a few taste buds on them, and Those little taste buds are all over the surface or the edges of them. And the last shape we're going to look at actually has several layers to it sometimes, and then a button in the middle. And all of these layers have taste buds on them. So these have the highest number of taste buds. Now when we studied ions, if you study ions and atoms in chemistry, you learn that the electrons travel in little layers or little orbitals. And the outermost is called the valence orbital. Well, valet refers to wall. And since these walls actually go around, if I were to look down on top of this, it would look like the button in the middle, there would be a gap, and then there would be a wall, a gap, and another wall. So they almost look like a target sign, so to speak. What we've done is we've cut it in half this way, and we're looking at it from this angle. This is the button in the middle. These two walls are going around. Because they have so many layers, there's more surface area for more taste buds. So these type of papillae have the highest number of taste buds, and they are called circumvallate papillae. Papilla singular, papillae would be plural. Circumvallate means walls around. Now, all these different papillae are located in specific locations on the tongue. But ultimately, what you need to know is that the filiform don't have any taste buds. So they're not functioning at taste. They do grip, and they can detect texture. This is what makes a cat's tongue rough, is that they can stick their tongue in a liquid, and the liquid sticks to these, and it pulls some of it in. Fungiform papillae have, 5, 10 or so. They have, maybe a dozen taste buds, quite a few. Folate papillae, similarly. And actually the folate papillae are located a little bit further back on the tongue than the fungiform, and then the circumvallate are near the very back of the tongue. And they have the highest number of taste buds. Okay, so you should know their anatomy. Now it turns out that the taste buds themselves can detect basically 4 primary taste sensations. There's some evidence now that there's a 5th, a 6th, and maybe even a 7th taste that we can detect. But we still refer to these as the 4 primary taste sensations, and then we'll add some of the other tastes. Our tongue actually has some geography to it. There's actually a thing called the geographic tongue. Some people who are sensitive to really hot foods, the papillae on their tongue can start to retract, and it looks like there's little flat or bald spots on the tongue, and then they come back. So it's an interesting feature of the tongue. So sometimes if you're working in healthcare and a parent's freaking out because their kid's tongue looks like it has some little flat spots on it where the papillae disappeared, they call that a geographic tongue. There is a geography to the tongue in a different sense, in that the location of these papillae is very specific. Okay, so if we were to look at a giant tongue here, you would realize that the 4 primary taste sensations we're going to list them, and then we're going to talk about some others. The 4 primary taste sensations. The first one is sweet. We have sweet receptors. There's 2 camps on this, and I want to explain something. It was originally thought by a guy who did a bunch of these studies. If I did the sweet receptors in green, the highest concentration of taste buds that detect sweet tastes are near the tip of the tongue. Okay. Now, some people thought that you only detected sweet at the tip of the tongue. That's not true. You have sweet receptors all over the tongue, but the closer we get to the tip, the higher in concentration they become. And so this part of the tongue is really, really sensitive to sweet. We can detect sugar all over the tongue, but we really have the highest concentration of sweet receptors near the tip. We're most sensitive there. The second sense that I'm going to talk about is salty. Salt receptors, those papillae, those taste buds that are sensitive to salt tastes, tend to be in the highest concentration over here on the sides of the tongue near the front. That doesn't mean we don't have any near the tip of the tongue or in other locations. You have salt receptors all over the tongue, but we do have the highest concentration in these areas here, and they'll overlap the salt receptors. Okay? That's why we like to combine sweet and salty sometimes together. A third taste sensation would be sour. Sour receptors are located further back on the tongue, at least in the highest concentration, way back here. and they will overlap some of the others. Again, that doesn't mean we don't have any sour receptors in other parts of the tongue. But if you're going to taste something sour and you put it on the tip of the tongue, it's less impact than if you put it back where the higher concentration of the sour receptors are. Some people believe, or it used to be taught, that we only had sweet here, only had salty there, only had sour there. And then some people realized, no, they're all over the tongue, and they blew that whole theory out of the water and said it doesn't exist. Well, it's actually somewhere between. We do have the higher concentration of sweet, salty, and sour in different locations. Again, we have them, each one of them, all over the tongue, but they are kind of locally concentrated. The last of the 4 primary taste sensations is going to be bitter. The bitter receptors are very far back on the tongue and occupy this area here. Okay. Doesn't mean we don't have bitter receptors in other locations, but when you taste something really bitter, it gets you on the back of the tongue, so to speak. So, now there are some other taste sensations that are not part of the 4 primary taste sensations, but when we want to taste foods, those foods that give us the greatest pleasure are those meals that combine have some combination and balance of sweet and salty and sour and bitter, so that we get a little bit of everything, and those are the meals that really, really, really speak to us. So we also have taste receptors for what they call umami. It's my understanding that the word umami is a Japanese word for broth, and what these can detect is the presence of protein, particularly the amino acid glutamate. The amino acid glutamate is abundant in a lot of proteins, and so, you know, if you ever watch survival shows, when people are starving, one of the things they complain about is the need for protein for the body. And so when we detect the presence of protein, it's important. We need to replace the proteins in our body in order to stay healthy. And so glutamate receptors, or umami, are very, very abundant, and they really are found associated with a lot of these circumvallate papillae near the back of the tongue. Okay. There are some people that believe we have water receptors that can detect the presence of water or moisture, and those are really way back here. Like when your throat's getting dry, when you become dehydrated, you crave watery stuff. And then there's some that detect peppery flavors. There's a protein called capsaicin that's present in peppers and jalapenos and things, and we can detect the sensation of heat or peppery flavors, but those really aren't considered the primary taste sensations. And there's some controversy as to some of these, but nonetheless. So, one of the things we do know is that the protein capsaicin, which provides the heat sensation when you do eat something really hot, really likes to bind to sweet receptors, and it'll really burn your tongue where all the sweet receptors are. That's why in order to dissipate the sweet, you need to drink something really rich in sugar to outcompete those capsaicin proteins for sweet. Anyway, that's just a little side trivia. So you need to know the 4 primary taste sensations. You need to have an idea of where they're located on the tongue. You also need to know this. If I divided the tongue into 3 major regions, the anterior 2/3 of the tongue are monitored by the cranial nerve number 7. And then the back of the tongue is monitored by cranial nerve number 9. So cranial nerves 7 and 9 are monitoring a lot of these taste sensations on the surface of the tongue. If we killed cranial nerve 7, you would lose a lot of sweet and sour taste. They would become less strong to you. If you destroyed cranial nerve number 9, then you wouldn't be able to detect some of these sensations. So no cranial nerves 7 and 9. If you go through the cranial nerves, remember it's olfactory, optic, oculomotor, trochlear, trigeminal, abducens, vestibulocochlear. It's olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial nerve, vestibulocochlear, glossopharyngeal. So the facial nerve and the glossopharyngeal nerve are monitoring the tongue. Sorry, I drew a blank there for a second. My son's walking through the house asking if we can go work out. So I got distracted for a moment. Anyway, now, when it comes to the anatomy of a taste bud, so let me do this last little bit and we'll stop here. We'll save the eyeball for another lecture. I'm going to erase all this information and we're going to actually look at an individual taste bud. Okay, so if I were looking at one of these papillae like this, and I had these little taste buds, these little pockets of cells on the side here, if I magnified one of these and looked at it, it would look like this. I would have this little opening, and I would have a little pocket that houses all these cells. That opening, by the way, is defined as a taste pore. Now, there are going to be some cells sitting inside the taste bud that are going to be long, narrow cells that look like they have these little hairs or cilia sticking off, and then they'll send their axons into the tongue. These cells, they're starting to look like olfactory receptors because they're very similar to them. And they'll stick their little taste hairs or cilia out of the taste pore so that they can bind to gustatory molecules. Okay, so when you taste something, a gustatory molecule will bind to those receptors, trigger a signal transduction cascade, and fire an action potential into your tongue, and then that would go out, those axons would go out one of the two nerves that we just talked about, cranial nerve 7 or cranial nerve 9. Now mixed in with these neurons are some cells in between them called support cells. So these purple cells that I'm drawing are support cells. And just like if you watched olfaction, gustation is only the second known type of neuronal replacement. These taste receptors, these gustatory receptor cells, will live about 10 days to 2 weeks, and then they die, and we're replacing them. So these support cells will grow up and develop into new gustatory receptors. They'll develop some cilia and an axon and rewire. And at the base are going to be some basal cells. The basal cells are highly mitotic. They are dividing by mitosis. The old cell stays behind. The new cell develops into a support cell, which develops into a gustatory receptor. So it's almost identical to olfaction, only we don't really have the lamina propria. That would be in here in the space of the tongue. Okay? So, know that there's two types of neuronal replacement that we know of in the human body in adults, which is olfactory receptors and gustatory receptors. And the sense of smell and taste are very closely linked. As a matter of fact, if you pinch your nose and you try to taste something, your ability to taste it, the intensity of the sensations, and the ability to detect different flavors is decreased. Our sense of smell enhances our sense of taste. That's why when you're cold and stuffed up or have the flu, not everything tastes good. Again, as we age, these gustatory receptors become harder to replace, and our ability to detect different aromas and different flavors declines as we get much older. As my professor once said, jokingly, this is why you can go eat at Luby's 5 or 6 days a week when you get older, and everything, tastes so great, and will really all just taste the same. Anyway, I'm going to stop there for gustation. I think we're done with that. I hope you learned something. I hope you had as much fun as I did. And I hope to see you on the flip side in the next video, where we're going to start talking about the anatomy of the eye and the physiology of the eye. Thanks for watching.